with lecture 13. This is going to be part 2 of the lecture. So in part 1, we talked about cell signaling and really got into all those wonky cell signaling pathways. And the cell cycle is really the next logical step in the cell because there are many pathways work together to regulate the cell cycle. Now, I will tell you, the cell cycle at this level in biology is very complex, and it can be a big challenge for students to learn. Like the pathways, it's a lot of information, a lot of different proteins. It can be difficult to kind of keep them all separate. And, there are a couple key figures in the cell cycle that if you don't have those down pat, understanding, the relationships between all the different cyclin-dependent kinases and cyclins, and how those work together to regulate the cell cycle can be really difficult. Okay, so just keep that in mind as you're working through this, and try not to get frustrated because it is tough. It's hard stuff, and it takes time. It takes practice and repetition to really fully understand it. Okay, now we're focusing on eukaryotic cell cycle. It's a very orderly sequence of events that allows a single cell to not just double, but it's part of the cell cycle is the cell's growth, then its DNA duplication, and then its ability to divide into 2 genetically identical daughter cells. It's the foundation of all cellular reproduction and tissue renewal. Its regulation really ensures that DNA replication happens only once per cycle, and that division occurs only after the genome has been faithfully and completely copied. So we're going to cover the first 2 sections out of 3 in the text. 17.1 is we're going to look at the 4 main phases of the cycle, how they've been experimentally analyzed, checkpoints and growth signals and things like that. 17.2, we're going to look more at the proteins that are involved in the different cycles, checkpoints. And then 17.3, which we're not going to cover, covers just M phase. And not that it isn't important, but it is definitely a more specialized subset, in my opinion, of cell biology. You can still understand cancer biology by just knowing the different stages and the checkpoints without focusing on M. And these 2 sections are going to be enough to challenge you. Okay, so let's get going here. So the cell cycle progresses through 4 distinct phases: G1, S, G2, and M. Okay, the letter G stands for gap, the letter S stands for synthesis, and M stands for mitosis. Now, some people say G stands for growth. Both are fine. Gap, growth. So during G1, the cell grows and prepares for DNA replication by producing the proteins it needs, the organelles. It's in its normal stage of life, really. That's what G1 is. Now, at some point, the cell makes a decision to go into the next phase. And now it's no longer in its normal stage of life. It is preparing to divide. And that is the S phase. And that's when the DNA replication occurs. It creates identical copies of every chromosome. Following that, G2, it adds more growth and prepares the cell for the next stage, which is mitosis. Finally, during M phase, both mitosis, which is a nuclear division, and cytokinesis, which is the cytoplasmic division, take place, ultimately producing 2 identical daughter cells. So, together, G1, S, G2, they make up interphase, the period of metabolic activity between divisions. In contrast, M represents the very short, dramatic events of how the chromosomes segregate and the cytoplasm and cells divide. So, interestingly, in early embryonic cells, the cycle lacks G1 and G2 phases. It consists of very rapid S synthesis, DNA synthesis phase, and mitotic oscillations that generate many smaller cells from just the large fertilized egg. And there's a reason for this. If you look, so I'm just going to jump over here, you have a fixed volume here, and this is going to develop into a blastula. So in order to do that, you don't need the other gap or growth phases to increase the cell size and whatnot. So during early embryonic development, it's just a cycling between M and S. Anyway, so over here, you've got the standard 4 phases of the division cycle, how it moves sequentially through G1, S, G2, M. It also is emphasizing how interphase occupies the majority of the cycle. Most of the time spent in growth and DNA synthesis, while M phase is relatively brief. G2 is briefer than these two sections, right? And we talked about embryonic cell cycle, right? All right, so progression through the cell cycle can be measured experimentally. So classically, the ways that it's done is Western blot. You can certainly detect cell cycle through Western blot by looking at the certain proteins that are expressed at certain stages. But, we've done a Western blot. It's kind of involved in terms of time. It takes a number of days. A very rapid test for cell cycle was developed some time ago after a flow cytometer, after the flow cytometer was invented. So in flow cytometry, cells are suspended in liquid. It's called a sheath fluid. And then they're basically propelled through a capillary system that lines the cells up one behind each other, so that you have just a line of cells. And it sends it through a flow cell where you have a laser that's shining in the cell, so you can excite some sort of fluorescent property that the cell now has that you've added to it. And then you can detect it. So the detector's on the other side of the flow cell, and it detects the excitation from the fluorescent molecule. So you can use a flow cytometer to very quickly and accurately detect cell cycle and a lot of information about the cell cycle. What you're ultimately detecting with this cell cycle assay is DNA content, because DNA content changes rapidly through the different phases of the cell cycle. Cells in G1 have a 2N DNA level, right? And that's going to end up forming one distinct peak. Cells in S phase have an intermediate value between 2N and 4N. You get a broad middle peak, not really, just called a region, not a peak. And then cells in the G2M display 4N, right? Because the chromosomes have duplicated, and you get a second peak. So with that more DNA, you're going to get more fluorescence, right? And you can also, you're not, with flow cytometry, you're not just looking at fluorescence, but you're also, the cytometer is counting the number of cells or events that you're pushing through the flow cell. So with that, you can get this nice little histogram where you've got two axes. So you've got the number of cells that have been analyzed here, and you've got the amount of DNA, or the DNA content, because of the fluorescence with the fluorescent dye that you've added. The most common one is a dye called propidium iodide. And, there's a brief step where you have to take your population of cells and you fix them in ethanol. It takes like an hour or two. And then you stain your cells and put them in the flow cytometer. So here, this is like, a population, a normal growing population of cells that are mostly in G1, some are in S phase, and some are in G2 or M, right? So this is like the G1, G2M, and your S phase here. So what you can detect with this is growth arrest, right? So, if we're trying to analyze if a chemotherapy chemotherapy agent is working, we could do that very easily and at different concentrations and different incubation times and things like that. So what you see if the shift from left to right is the G2M is reducing. All right, so there are less cells, because I want you to look at this number here. So this axis goes from 700 to 1500. And here we've treated with either we've pulled serum from the media, or we've treated with something that induces growth arrest. So now all of our cells are arrested in G0, G1. So just a little something about G0, because we haven't talked about that. G0 is designated the quiescent stage, and sometimes it's permanent, sometimes it's temporary, but it's possible that cells have entered that phase. It's flow cytometry, PI staining cannot detect a difference between G0, G1, just like it really can't detect a difference between G2M. But in this one, it's been growth arrested because this peak has, even this sample has been a little growth arrested. It's got a very poor G2M peak, but this has been, completely, the cells have completely switched over to G0, G1. Okay. All right. So, using in experiments analyzing growth arrest, you can analyze these different phases. The model organism that was used to really elucidate the cell cycle initially was the yeast. And it's, you can't use bacterium. It doesn't have a cell cycle. It's prokaryotic. The yeasts do. And although it's a little different in yeast and the way they divide is a little different, their cytoplasm doesn't divide. They bud their yeast. You can see in this micrograph over here, you can see the yeast popping out. There's a new bud diverging. This one's producing two. It looks like udders. What was found in the yeast is that the G1 phase serves as a really critical decision-making stage that integrates both internal conditions and also external growth signals. And in yeast, the control point is called start. Right? And that control point is monitoring nutrient availabilities, it's monitoring the size of the cell, it's monitoring mating factors as well. Passage through start commits the cell to another cycle of replication and division. So in animal cells, there is an analogous checkpoint, and it's called the restriction point, or R point, and it occurs in late G1. Right? So here, growth factors determine whether a cell proceeds into S phase or exits into a quiescent non-dividing state known as G0. But once past the R point, progression to DNA replication becomes irreversible. Right? So these control mechanisms really ensure that cell division only proceeds under favorable conditions. And I apologize, I jumped right to the figure, so it's the same thing though. So this is the figure, yeast cell cycle, division's regulated by start, nutrient mating cues are what control this checkpoint. Budding begins immediately after start, you can see. So we're getting ready to go, and we got that micrograph over there. Okay. All right. So this figure is really illustrating the restriction point in animal cells. All right, so growth factor availability during G1 determines whether cells continue or enter G0, right? So if growth factors are pulled, you pull the serum in a cell culture, cells will enter this quiescent stage, and they stop dividing. It's a reversible pause that can last days, months, or even years. All right, cell cycle checkpoints. Cell cycle checkpoints are provided because they preserve genomic integrity, right? And they use these checkpoints to control mechanisms that monitor and coordinate the cycle's progression. They really act like molecular surveillance systems, and they do that to ensure that each phase is correctly completed before the next phase begins. So there's the G1 checkpoint. This is a DNA damage checkpoint, and it prevents entry into S phase if DNA is damaged. The S phase checkpoint detects replication stress or stalled forks. The G2 checkpoint prevents entry into mitosis until DNA replication is complete and damage repaired. And then finally, there is a spindle assembly checkpoint, which is during M phase, that ensures chromosomes are properly attached to the spindle before anaphase begins. If any checkpoint fails, the result can be mutated DNA, the result can be aneuploidy, or the result could be tumorigenesis. So, these checkpoints ensure that only intact, fully replicated genomes are passed to daughter cells. Now, the spindle assembly checkpoint, we're not going to cover a lot of information on that, and that's okay. That's really covered in the next section, 17.3, which we're not going to do. Okay, so this is an important figure. There's a figure that's like this that we'll see in a little bit that's even more important, but this kind of shows you the checkpoints and where they are relative to the cell cycle. These are the major surveillance points that govern cell cycle progression. You can see G1, S, G2, and M phase, and then each associated with these control processes. So these DNA damage checkpoints detect unreplicated or damaged DNA, and they halt progression until repair is complete. The spindle assembly checkpoint will arrest mitosis if chromosomes are misaligned, and if they're misaligned, that could cause an unequal segregation. Right. So, the cell cycle is not just a simple timer. It's a quality control system as well. All right. So in 7.2, we're going to get into some more challenging content. We've just seen how the cell cycle follows a precise sequence of growth and division. But what actually drives those transitions at the molecular level is what we're going to explore in this next section. So we're going to focus on two key regulators. They are called cyclin-dependent kinases, or CDKs, and cyclins, all right? And they're partners of each other. So these two key regulators serve as like these biochemical engines of cell cycle progression. Each phase of the cycle is triggered by a specific CDK cyclin complex that activates the enzymes and structural proteins needed for that stage. So together, CDKs and cyclins ensure that the cell moves forward in a very orderly fashion, growth, replication, and division, and never backwards. And where we're going to start out is looking how discovery began with identifying a single very powerful regulator called MPF, or mitosis promoting factor, or maturation promoting factor. And it was initially done in frog eggs. So this was the first evidence of a universal cell cycle regulator looking at frog eggs. So normally, G2 arrested oocytes require the hormone progesterone to enter M phase. However, early on, when scientists injected the cytoplasm from M phase oocytes into G2 arrested ones, the recipients entered mitosis without progesterone stimulation. So what did this tell you? Well, this demonstrated the existence of some sort of cytoplasmic factor, which was later named maturation promoting factor, that was sufficient to induce the G2M transition. Biochemically, MPF is found to be a complex of CDK1. Right? Now take apart that cyclin-dependent kinase, right? Here's a protein that depends on cyclins to function, and it is a kinase. It's a protein that can phosphorylate other proteins. So MPF was found to be a complex of CDK1 and cyclin B, right? Cyclin is the CDK's regulatory subunit. Together, CDK1 and cyclin B trigger mitosis by phosphorylating proteins involved in the breakdown of the nuclear envelope, the formation of the spindle, and chromosome condensation, right? All those things needed are in prophase of M phase. MPF activity rises and falls cyclically as cyclin B accumulates during the cell cycle and is later degraded, revealing a molecular rhythm of the cell cycle. Right here, this is what happened to do a cytoplasmic transfer in xenopus oocytes. So here, the donor oocyte, which has already transitioned from G2 to M phase, provides cytoplasm that's rich in active MPF. When the cytoplasm is microinjected into a G2 arrested recipient oocyte, the recipient immediately enters mitosis, even without progesterone stimulation. So this experiment proved that MPF activity is cytoplasmic and transferable, and established the principle early on that cell cycle transitions are driven by regulators, not membrane-bound signals. And it paved the way for identification of the CDKs and the cyclins. Right, but the real work and identification of all the CDKs and the cyclins began that were behind MPF began in yeast. So genetic studies early on of temperature-sensitive CDC28 mutants in yeast showed that these cells arrested at start when grown at restrictive temperatures, meaning that CDK1, which is the protein that's encoded by CDC28. So CDC28 is the gene that codes for the cyclin-dependent kinase 1. So since these cells were arrested at start when grown at different temperatures, we know that CDK1 is essential for triggering cell cycle entry. So it was found that CDKs are evolutionary conserved across all eukaryotes. And then, so that's discovered the CDKs, studies in yeast. And around the same time, studies in sea urchin embryos revealed another family of proteins called the cyclins. And they accumulate during interphase and disappear rapidly after mitosis. They have oscillating levels that parallel the activity of MPF. This confirms that cyclin synthesis and degradation are what serves as the timing mechanism for the activation of CDKs. So the CDK1 cyclin B complex became really reorganized as the universal mitotic entry system. Right. So here we've got the phenotype of temperature-sensitive CDC28 yeast mutants, right? So at non-permissive temperatures, you were getting arrested start, but at permissive temperatures, it was going through. And the reason for that was at the permissive temperature, you were able to produce CDK1. It's basically CDK1 function is restored. Over here, we've got the sea urchins, right? You see the cyclic pattern of cyclin accumulation and degradation. In sea urchin embryos, cyclins build up steadily during interphase and are abruptly destroyed at the end of mitosis. And this destruction is crucial because it resets the cell cycle machinery. And that ensures each division event begins anew, right? So, these two figures here really capture the logic of the cycle, right? You have a stable kinase engine, CDK1, and regulated by a fluctuating control protein, which is cyclin B. All right, so it was eventually discovered that MPF is, what is MPF? Well, MPF is a heterodimer composed of CDK1, the catalytic kinase, and cyclin B, the regulatory subunit. Cyclin B binding is required for CDK1's activity. It not only activates the kinase, but it also targets it to appropriate substrates within the nucleus and cytoplasm. Once active, MPF phosphorylates then multiple proteins that collectively execute mitotic entry, such as condensation of chromatin, disassembly of the nuclear envelope, and formation of the mitotic spindle. The system is also exquisitely controlled by reversible phosphorylation, and that's real important in the timing as well. Here you can see the structure of MPF. It's actually composed of CDK1, which carries the kinase activity. It's the catalytic part of the heterodimer. And the other is cyclin B. The association of cyclin B is necessary for proper folding and activity of CDK1. Neither subunit alone is functional. Only the dimer acts as the active trigger for mitosis. So the pairing between CDK1 and cyclin B is like a universal template. All CDKs and cyclins behave in this manner. They have to pair up. All right, so CDK1 activity is controlled by a precise balance of phosphorylation and dephosphorylation. During G2, okay, we're starting here at G2. So we've done G1, we're doing M, we've done S. I'm sorry, we did G1, we did S, and now we're on to G2. So during G2, CDK1, it binds to cyclin B, but it's inactive. Right? So it's inactive MPF complex. So there's an enzyme called CAK, C-A-K. It stands for CDK activating kinase, and it phosphorylates CDK1. Right? It does it specifically at these two residues, threonine 161, and just at one residue, threonine 161. All right. There's another kinase called WEE1, which adds inhibitory phosphates to the CDK1. So here we are in G2. We've got our cyclin B and CDK1 complex, and now we're loading it up with phosphates, and we're using two enzymes to do it. One is a phosphate that's going to add inhibitory phosphates, and that's WEE1, and one that's going to add activating phosphates, and that's CAK. Okay. So after you've loaded up with phosphates, there is a phosphatase that can remove the two phosphates that were added by WEE1. This is called CDC25, right? CDC25 phosphatase that removes the inhibitory phosphates, and now we have an active CDK1, and that triggers mitosis. So it's not just the cyclins and the CDKs. It's a bigger story. There are phosphates and phosphatases that are involved in the final activation. But obviously the pairing up of CDKs and the corresponding cyclin is very important. All right, so after it's been activated, then we know it's going to have to be deactivated, and that's going to occur after mitosis. So at the mitotic exit, cyclin B gets ubiquinated, and it gets ubiquinated by a complex made up of APC and also C protein. Okay, so it's anaphase promoting complex, and C is called the cyclosome. It's a regulatory protein, and APC and C, they start sticking all these ubiquitins on cyclin B, and that targets it for degradation by the proteasome. Okay, that inactivates CDK1 and resets the cycle. Okay, so here we can see it going on. All right, so we started over here. Cyclin B and CDK1 pair up, and they get loaded with phosphates, both from CAK and WEE1. CAK is adding the activating phosphate, and WEE is adding the inhibitory phosphate. So it's still inhibited. Finally, a phosphatase, CDC25, right, pulls the inhibitory phosphates off, and now we have an active MPF, and you get into mitosis. Following M stage, a complex of anaphase promoting complex and the cyclosome pair up, and they dephosphorylate. I'm sorry, they target cyclin B for degradation. Okay. CDK1 is dephosphorylated. Eventually, some more cyclin B is made, and then you have the turning of the cell cycle. Okay, so yeah, this is a big important control step. Transitioning into M phase. All right, so that's cyclin CDK1 and cyclin B. But there, of course, it's not that simple. There is a family of cyclins and CDKs that are each assigned to specific phases of the cell cycle. And this is where, for most students, this stuff starts to get overwhelming. And try to bear with me. I've got figures that I'm going to keep up on the slides because in case you forget which cyclins are involved as we get through this material. But let's start going through it. So in G1, CDK4 and CDK6, right, both pair, not with each other, but with D-type cyclins, or cyclin D. And it controls passage through the restriction point, right? That's in G1. The next thing is the G1S transition. At the G1S transition, it's CDK2 and cyclin E that initiates DNA replication. Now we're in S phase, right? In G2, CDK2 and cyclin A maintains replication and prepares the cell for mitosis. Finally, CDK1 and cyclin A and B are what drive the mitotic events. Okay, so this sequential activation ensures an irreversible ordered progression through the cell cycle. Later cyclins help inactivate earlier ones, and that enforces directionality. There are also proteins in the cell that act as CDK inhibitors, and they can provide negative control when conditions are unfavorable. This is a very important figure that you need to learn. And at some point, it's a kind of mix of just plain old memorization and also understanding. So there's some mnemonics that can kind of help you learn it. You should be able to draw the phases of the cell cycle from memory. Understand that there are transition points between every step except from M to G1. You have a transition point past the restriction point, right? You have a transition that goes from before R and after R. You have a transition point that goes from G1 to S. You have a transition that goes from S to G2. And you have a transition that goes from G2 to M. There's also within M a complex that drives it through. But there's no transition from M to G1, right? So because at the end of M, you have two daughter cells that are in G1. So there are cyclins and CDKs. To learn the cyclins, it's D, E, A, B in this direction, starting here in G1. Okay, they've got decision, entry, advance, breakdown. Right, that's what I've looked up today. That's how the kids are learning it today. When I was in school, we learned drug enforcement agency is BS. Okay, that's what we learned in school. Whatever. All right, so that'll teach you with there you can kind of figure out the order of the cyclins, D, E, A, B. And then for the CDKs, it's 1, 1, 4, 6, 2, 2. Okay, and you start here up at M. It's 1, 1, 4, 6, 2, 2. And if you look, these CDKs have like common roles. Like CDK2 has involvement in DNA. CDK1 has involvement in cell preparation and getting ready for M. CDK4 and 6 has to do with that restriction point. Okay, so with some practice, you will be able to memorize this, but really knowing this is going to be crucial for understanding the rest of the section, because now we're going to start looking at regulation of these different complexes. So do what you have to do to learn it. So as we're moving forward, I'm going to keep this figure on here just so you can refer to it. So we know it's doubtful you've just memorized that. So that's going to be a reference figure moving forward. Okay. All right. So this table is listing those CDK inhibitors that I was talking about, called CKIs. You don't need to memorize these, really the families. P21 and P27 are the most prominent because they're implicated in disease. They have roles that we're going to talk about. These are not chemical inhibitors, but cellular proteins, and they can act as brakes when DNA damage or unfavorable conditions are detected. Like cyclins and CDKs themselves, they are regulators of cell cycle control. but think of them as only being in play under abnormal conditions. Okay, so it's the CYP-KIT family that are more prevalent. They're involved in both G1 and SG2, and you know, moving forward. Okay, all right. So this slide is going to show how growth factors initiate G1 progression through cyclin D synthesis. Okay, so we're making cyclin D here. So where are we in this thing? Well, we're getting past, we want to get past the restriction point. We're talking cyclin D. So the pathway we learned about in the previous chapter, the RAS, RAF, MET, ERK pathway, the MAP kinase pathway as it's called, responds to mitogenic signals, right? So mitogenic signals are signals that induce mitosis, like growth factors. When mitogenic signals activate the RAS, RAF, MEK, ERK pathway, ERK moves into the nucleus and activates the ELK1 serum response factor transcription complex. Okay, we talked about that in the last chapter. That ultimately drives transcription of D-type cyclins. That's one of the consequences of activating that pathway. Cyclin D then binds to CDK4 and CDK6, forming active complexes that begin phosphorylating cell cycle substrates. Okay? These events are preparing the cell to cross the restriction point. Okay? So there's kind of listed out. You get growth factors, activation of the MAP kinase pathway, activation of ELK1 and serum response factor, and expression of AP1 transcription factors, and ultimately synthesis of cyclin Ds. Okay, cyclin Ds can bind to CDK4 and CDK6, and then these guys can get us past the restriction point. Okay. All right, so that is the all-important MAP kinase pathway. Now, there are two other proteins called RB, which comes from retinoblastoma protein, or retinoblastoma, which is a cancer in the eye. And it was discovered through discovering these cancers in the eye. And then there's another protein called E2F, which is a transcription factor. So these two interact, RB and E2F, and it's like another regulatory axis. And these proteins are now coming up in the cell cycle, and we're going to revisit them when we go into the next chapters, talking about oncogenes and tumor suppressors, because these are potent oncogenes and tumor suppressor proteins. All right, so you have RB, and in its unphosphorylated form, it will bind to ETF, right? And ETF is a family of transcription factors. It will bind to them and repress the genes required for S-phase entry. Right? How these proteins interact with each other is what permits a cell to go into S-phase or not. As CDK4 and 6 and cyclin D complexes become active, they will phosphorylate RB. That causes it to come off of E2F. When E2F is free as a transcription factor, it will stimulate transcription of cyclin E, CDK2, and genes involved in DNA synthesis. Stimulating cyclin E and CDK2, you can see, is getting the next transition done, G1 to S. Okay. So, loss of RB or overactivation of CDK4/6 cyclin D removes the restraint, right? So, and that is a hallmark of many cancers. Okay, so here we can see how RB phosphorylation controls S-phase entry. All right, and in G1, RB binds to E2F and it silences its target genes. Then, upon phosphorylation of RB by CDK4/6 cyclin D, RB changes conformation and releases E2F to activate transcription. The result is the synthesis of cyclin E and CDK2. That establishes the next layer of control, and that part is not shown in the figure here, but that's what RB is. That's what ETF is doing right now. It's synthesizing, it's driving the transcription of cyclin E and CDK2. So RB and E2F is the most important toggle switch in cell cycle control, as it's the first switch, right, going from G1 to S. And because of that, these proteins are pretty heavily implicated in cancer as well. All right, so now we're going to talk about E2F-driven activation of cyclin E. All right, when E2F induces the cyclin E and CDK2 complex to form, that will further phosphorylate RB, creating a like a self-reinforcing loop. Okay. Early in G1, the CDK inhibitor P27 will restrain this complex, but growth factor signaling lowers P27 levels. Active CDK2 then phosphorylates P27, targeting it for degradation. The resulting surge in CDK2 cyclin E activity inhibits the APCC ligase, preventing cyclin E destruction and ensuring irreversible passage through the restriction point. So interestingly, overactivation of cyclin E or loss of P27 will remove this control. That is also a very common feature in uncontrolled tumor proliferation. Okay, so here we've got a figure that is showing activation and inhibition. All right, so starting at the top here, we see how you got P27 binds and inhibits CDK2 and cyclin E. Okay, and this is early in G1. Passage through the restriction point induces cyclin E, and we now know that happens via E2F. And so passing through the restrictions point induces cyclin E and stimulates suppression of P27 synthesis through growth factor signaling. Okay? Once CDK2 becomes active, as shown there on the right, with the yellow highlight there, it phosphorylates P27, marking it for proteasomal degradation. So now you have a whole bunch of active CDK2 and cyclin E, and you can progress from G1 to S. All right, so this is a feedback loop that amplifies, like very rapidly, CDK2 activity and also greatly inhibiting APC and C. Inhibition of APCC prevents premature cyclin degradation, and it locks the cell into S phase commitment. All right. So here we're going to talk about how CDK2 and cyclin E initiates and regulates DNA replication. Okay. So CDK2, cyclin E is going to initiate DNA replication, and basically rising levels of this complex activate replication machinery. One in particular, if you remember back from our studies of DNA replication, there is CMG helicase complex that unwinds the DNA replication origins. So rising levels of CDK2 and cyclin E will activate CMG helicase. After CMG helicase unwinds DNA replication origins, DNA polymerases are then recruited to begin synthesis. And this is important. Once replication begins, CDK activity prevents the reassembly of new replication initiation complexes at the origins. This ensures each DNA sequence is copied once and only once per cycle, right? So if you prevent new origin initiation complexes from growing up, you can't replicate again. So this guarantees real faithful genome duplication and prevents genome stability. All right, let's look at oops, did I skip a thing? I skipped something here. Okay. Yes. No, I didn't skip anything. All right. So on to ATM and ATR here. So specifically the DNA damage detection and checkpoints. So how is DNA damage detected? The cell cycle includes these built-in safety systems that monitor DNA integrity. And two proteins in particular, ATR and ATM, which are both kinases, can detect different types of damage. ATR responds to single-stranded or replicated DNA or unreplicated DNA, while ATM responds to double-stranded breaks. Once they're activated, they phosphorylate and activate two other proteins called CHEK1 and CHEK2, which are kinases. And they do it respectively. ATR works with CHEK1, ATM, CHEK2. These checkpoint kinases then phosphorylate and inhibit CDC25 phosphatases. Remember back from CDK1, preventing activation of CDK1 and CDK2. The result is cell cycle arrest until the DNA is repaired. All right, this arrest is really important because this checkpoint system allows for correction, right? So the arrest stalls the system so that the DNA can be repaired. All right, so here we're looking at how ATR and ATM are like, like sentinels of genomic stability, really. The DNA damage recruit sensor complexes will activate these kinases. So ATR activates CHEK1, ATM detected its double-stranded break, activates CHEK2, and these guys inhibit CDC25. Remember, CDC25 is a protein phosphatase, blocks dephosphorylation and activation of the CDKs. This halts the cycle at the appropriate checkpoint until repair is completed. All right, last thing here, we're going to talk about protein P53, and very appropriate to talk about it now, as this is going to gateway us into the next section, which is cancer and apoptosis. It's a very important protein, P53. It's also called like the master guardian of the genome. And under normal conditions, P53 is constantly ubiquinated by MDM2 and degraded. So it's like the gene is translated and the protein's broken down under normal cellular conditions. And just as a side note here, this is why proteasome inhibitors are very effective cancer treatments, because you inhibit the proteasome and now P53 levels build up. And anyways, we're going to get back into that, but it's because P53 is a tumor suppressor protein. It's a protein that causes cell cycle arrest. It slows down the breaks, slows down the cell cycle. So when DNA damage occurs, ATM and CHEK2 phosphorylate P53, preventing MDM2 from binding and tagging with ubiquitin. Stabilized P53 now accumulates in the nucleus. It activates transcription of P21. This is a CDK inhibitor, thus arresting the cell cycle. P53 is a lot of things. Transcription factor, it has some very important roles here. Just another thing to note, CDK2 inhibitors are a very new class of chemotherapies. They're under very heavy investigation, clinical trials. CDK4 inhibitors are already in the clinic. They have very strong efficacy, but CDK2 compounds could be really wonderful chemotherapies. They could really have fewer side effects than like the current cisplatin, doxorubicin, and paclitaxel cocktails, because they're not DNA damaging, right? To microtubule stabilizing. So, anyways, increasing levels of P21 binds and inhibits CDK2 and cyclin E. Very effectively by halting cell cycle progression at the G1S or S phase. All right, so this pause provides time for DNA repair, but maybe the damage is not repairable. P53, sustained P53 activity actually shifts the response from arrest to apoptosis. And this is very important to eliminate potentially cancerous cells. P53 mutations are found in many, many cancers. And its involvement in both cell cycle arrest and apoptosis is what truly makes P53 like the guardian of the genome. Okay. All right. Last slide in this lecture. You can see here in undamaged cells, MDM2 keeps P53 levels low by tagging it with ubiquitin and target it for degradation by the proteasome. However, as the cell accumulates DNA damage, right, phosphorylation by ATM and CHEK2 disrupts the process. Right, phosphorylated P53 can't be ubiquitinated by MDM2. And P53 then induces expression of P21, and P21 binds CDKs and stops the cell cycle. Okay, CDK2 and cyclin E. All right, so we looked at how the cell cycle works, and then also a bunch of different ways of regulating it, through the brakes of E2F and RB, and the cell cycle arrestor, which is P53. Okay. All right. That wraps up this very challenging lecture 13. And we will continue next time with the rest of, this is getting close to the end here. So pretty much one of the final lectures, if not the final lectures. Okay. All right, guys. Hope everyone's doing well, and I will talk to you soon.